Hey everyone, welcome back to my channel. My name is Naisargi and in today's video we are going to discuss about the methods for safe disposal of the biohazardous wastes that are produced during our experimentation in a biology laboratory. So till now we have discussed about the safe working methods in a biology laboratory which ensures the safety of our environment as well as the working personnel, in addition to maintaining the integrity, the quality and the authenticity of the experimental or the scientific data. But what happens after we are done with our experimentation? Whether we are working in an animal house facility, cell culture facility, microbiology lab or a routine biology lab, after we are done with our experiments, there are a lot of wastes which are produced and there are protocols for the safe disposal of these wastes. Often we have this approach that okay, now we are done with our experiments and what happens next is none of our concern. But this is not the correct approach. For example, if we are working with any cancerous cell line and we want to throw away any contaminated flask, we can just not dispose of directly in our dustbins or in the sink, because this will cause the spread of that cancerous cell line or the bacteria or the fungus which is the contaminant in that particular flask. This is just an example, okay? What I mean to say is that we cannot be that selfish and we cannot be that ruthless to the environment or to the other people who are not working in the lab. So that is why the safe disposal of our biological waste should be a mandatory practice. You might be following a simple routine of keeping the solid as well as the liquid discard beakers separate while you work in a biosafety cabinet in a cell culture laboratory. This is a very good example of mindful discard of the biohazard waste products at a smaller scale. So in this video, what we will be discussing: first, the disposal of the animal house waste products, the disposal of the products that are contaminated with blood, the disposal of routine plasticware, the disposal of routine stationery, the disposal of the eukaryotic cell culture lab components, the safe disposal of the bacterial cell culture lab components, and the routine biology laboratory wastes. Firstly, the discard protocols for the waste produced in an animal house. What are the wastes that are produced in the animal house? If you are working routinely in an animal house, you might be aware about these waste products that I am talking about. These include the dead animals, the treated animals, the animals which died because of natural causes, the animals which died because of the disease, the carcasses of the animals that you dissected, the plasticware, the tissues, the gloves, the caps, all of these are also contaminated with the animal blood or animal tissues. So these animal house waste can be categorized into two types: decompostable and non-decompostable. So the decompostable waste include the dead bodies of the animals or the discarded animal tissues. So for this, generally the practice of incineration is observed in an animal house even if your animal is not deceased as such. If these dead tissues they keep lying around or if they are discarded just like that, they may be the potential cause of the spread of various diseases. Hence it is very much important that you incinerate or burn these animal tissues for their safe disposal. The other wastes of the animal house, as I mentioned before, which include the plasticware or the disposables like gloves, caps, etc. that are contaminated with animal tissues or blood, they are disposed of in a special dustbin provided in the animal house, and then the waste of these dustbins is collected timely by the collection trucks for their safe disposal. Next comes the disposal of the waste produced in a bacterial cell culture laboratory, or as we call it, microbiology laboratory. So here we practice a system known as decontamination cycle. In this decontamination cycle, we collect all the used media which are contaminated by various bacteria or other microorganisms. We collect it and then we autoclave it so that all the microbes in those media get killed, and after that we safely dispose of the decontaminated media. These are then picked up by appropriate chemical waste disposal service providers. You can simply not discard these used media directly in the sink or your dustbin because there is a lot of bacteria bacterial or any other microorganisms growth in those media, because of course they were used for that purpose on the first place. And if these microorganisms they spread, they may be the potential cause of various diseases. Next is the protocol for the safe disposal of eukaryotic cell culture labs. So what are the wastes that are produced in these eukaryotic cell culture labs? The used medium, used buffer solutions, used drug solutions, the contaminated cell lines, and the usual waste, the used tissues, the used plasticware, the used gloves, the mask, and so on. So here also the general waste, that is the tissues or the plasticware, etc., those go into one dustbin. Now these dustbins waste, they are collected by the waste disposal management trucks on a timely basis. Suppose you are performing any experiment or changing the media or passaging or splitting your particular cell line, you will of course produce a lot of liquid waste. So the liquid discard of these labs, they include the discarded media, the contaminated media, the contaminated cell lines, and so on. So these liquid discard you have to collect in a separate discard beaker while you are working, and after you are done working, you have to add hypochlorite solution, that is the sodium hypochlorite solution to it. You have to treat these liquids with sodium hypochlorite for minimum 24 hours, and after that you can throw it in your discard bin, which is collected by the chemical waste disposal system trucks on a timely basis. Now the components that are contaminated with blood, that may include your capillaries, your syringes, your lancets, your vacutainers, etc. So prior to the disposal of these components in the respective waste bins, they need to be incinerated for the complete removal of any pathogenic substances. Special incinerators are available in the market for the incineration of these components prior to their disposal in their respective waste bins. For any of these biological waste, there are separate waste bins which are kept in every laboratory, and these dustbins are color coded. The image of these color coded waste bins I am going to insert in this video, so you can check for your reference. So remember that whenever you are discarding any of these biological components, you need to follow these protocols, and after following these protocols, you have to discard those components in the respective color coded waste bins only. And these waste bins, they are picked up by various waste management trucks. So this is all for today's video. I hope it was useful for you guys. Even if it was not useful, please make sure that you follow these protocols and work mindfully and wisely in your biology laboratory. So I hope by now you have clarity regarding how to work safely in your laboratory, ensuring your as well as your environmental safety. So this marks the end of my biosafety video series. In this series, I have made total 3 videos. The first video was about ensuring your own safety, which is by ensuring the various good laboratory practices. The second video was about biosafety levels by which you ensure the safety of the environment as well. And the third video was today's video, in which we learned about the protocols to ensure the safe disposal after our experimental work is over. I'll soon come up with my next video. You guys till then keep spreading kindness and keep spreading knowledge, as I always say, because this world needs a lot of it. Bye bye.